Okay, folks, welcome back. This is the second lecture in the cancer theme. Important lecture, really, to understand cancer. A lot of the mysterious things about cancer, sort of why can't we cure it? How can you both inherit cancer from your parents, and how can things like smoking cigarettes also cause cancer? Really, we need to understand the materials that are in this lecture. Cancer is a genetic disease. It's a disease that always involves DNA, right? And we always have to have DNA. We've said right from the start, one of the sort of defining characters of life is this ability to use information encoded in our DNA. And so as long as that's the case, we're going to have cancer. In fact, I saw just last night a study that found a bone from a dinosaur, and that fossil suggested that that dinosaur had some type of malignant cancer. So it's a very ancient disease that's just sort of a byproduct of being alive. And so, okay, well, let's go ahead and get into this lecture. I'm looking at the study questions. All right, so we're starting out chapter 4. A little confusing because we jump different chapters, right? The textbook was really not written for a cancer theme, but I've kind of pieced it together with this reading list and the lectures. So that's one of my goals in lectures, to make sense and make the reading list into a kind of a cohesive story or a narrative. So first question here is, describe the flow of genetic information in a cell. What molecule contains the information? Okay. And so here is the first slide, and it's showing really the DNA contains that information. Okay, and we do see a flow of information from the DNA, and then the end product is a protein, and along the way we have this RNA molecule, right? The DNA contains the information. The mRNA is just a temporary, relatively short-lived copy of that information. The DNA always stays inside of the nucleus. Okay, the nucleus has a membrane. The membrane around the nucleus here, the nuclear membrane, is actually the same type of information, or the same type of material rather, that forms a plasma membrane around the outside of the cell. Okay, so that nuclear membrane keeps the DNA inside, right? So information is in the nucleus. The information is used in the cytoplasm by the ribosome, and then the information is used to build proteins. Okay, and by the end of this lecture, you should have a better understanding about the big deal with proteins. Okay, so again, we start DNA, make a copy called messenger RNA, which leaves through a little pore in the nuclear membrane and goes out to the cytoplasm, where it is read by the ribosome to make a protein. Okay, and so that's really biotechnology, vaccines, virology. All of this one diagram really contains some of the most important information to understand a lot of biology. Okay, describe the three-dimensional shape of the molecule here being DNA, and that is a double helix. Okay, you can see in the lobby downstairs in Dobbs, the double helix is laid out on the floor. It's a long molecule, so within one of your cells, and you have trillions of cells, you have up to a meter or more of DNA packed into that one tiny little cell, 100 micrometers. There are two, it's called the double helix because there's two kind of backbones, here shown as blue and yellow, that run parallel to each other and form this helix, and then going between the two backbones are these things called they're called nucleotide the bases, right? Here shown as A's, T's, C's, and G's. Okay, let's see what the next slide. So here the blue and the colors are maintained. The yellow and blue shown on the far left and the far right is the same blue and yellow shown here as those kind of the sides of the ladder. So if you took this double helical molecule and you stretched it out and straightened it out, basically you'd have a ladder with the blue and yellow sides of the ladder, and then the bases as the rungs going between the sides of the ladder. Okay, now those the bases do pair with each other. So A is an adenine. It's an individual discrete base, and it always pairs and bonds to a thymine. That's shown as the T here. Or A pairs with T. C always pair with G. Okay, and so that forms the rung of the ladder. Okay, so again, we see the same thing. Should be able to see the parallels. The blue and the yellow are actually phosphates and sugars. So there's a sugar phosphate backbone, and then the bases here, A's, T's, C's, and G's pair across the middle of this molecule. And keep in mind too, the other thing is that they are anti-parallel strands. So the strand on the left is running one way, right? And we can see that terminal phosphate is on the top on the left side. The terminal or end phosphate down here is on the right side, on the bottom. Okay, so this one is running this way, this one is running that way. And where is the information? Okay, again, really DNA is one of the most important molecules in our cell, but it's actually just a physical molecule. The reason it's so important because of the information contained in it. That information, the sequence of A's and T's and C's and G's, is really what makes us unique as individuals. And we see, really, obviously, the information can't be contained in the backbone, that sugars and phosphate, because there's no variability there. It's just a sugar, a phosphate, a sugar, a phosphate. The variability comes in the bases in the middle. So the genetic code here, and humans have now sequenced our entire genome, and we know that that's just a library of trillions of A's and T's and C's and G's, right? So that's where the information is contained. Okay. So again, here's the double-stranded DNA shown kind of stretched out, straightened out, looking at the individual molecules in this diagram. The DNA is shown here again with even less detail. But we see that double helix here, and across, not shown in the diagram, going across between those two backbones would be the information, those A's and T's and C's and G's. Okay, so how does the cell use that information? And that is, in order to understand that, there's this thing called the central dogma of molecular biology. Okay, and so it's important that you understand the tenets of this, right? A real basic level. If you're a bio major, you're going to get into very specific details of this. And if you like that, you could maybe have a great career in biotechnology, right? So DNA, the information always stays in the nucleus, right? And then we have this process called transcription that makes our go from a DNA to an RNA. And when you transcribe something, if you think about these scribes in ancient times, that's books where we didn't have computers and printing presses. If you wanted a copy of a book, you would have somebody transcribe. They would look at this book and they would rewrite exactly the same information in the same language into another book. So that's really what's happening here. We're just it's a little bit oversimplified to say that we're writing A's and T's and C's and G's, because there is some differences, tiny differences, but suffice to say we're taking the information coded in those bases and making another copy of it as an RNA molecule. We send that RNA molecule out to the ribosome, and now we are translating from A's, T's, C's, and G's into protein language. Okay? So transcription happens in the nucleus. Translation happens in cytoplasm. Transcription, copying DNA to RNA. Translation, we're going to take the information that ultimately came from the DNA and we're going to use it to make a protein. Okay. Now, kind of a lot going on in this diagram. Figure 10.9. You should spend some time with this one. Read over the description of it and understand. Okay, so we see at the top our DNA molecule. Same molecule. Now, a new piece of information here. This DNA molecule is one of your chromosomes. Say we have 23 different chromosomes. Maybe this is chromosome 14. On that chromosome, there are multiple genes. Okay, I know you've heard of chromosomes. You've heard of genes. The relationship between the two. Chromosome is a long strand of DNA, and gene is just one segment of that chromosome. What does a gene do? Just information. But it is specifically the information needed to code for one polypeptide or protein. Okay. So information is up here. Again, during transcription inside the nucleus, the cell opens up, exposes one of the strands of DNA, and along that the information here is A's and C's and G's in this case. Transcription, again, we're making a copy. Every time there's an A, we write a U. Now, so again, here's one of the small differences. We are using the same language, but in RNA molecules, there's no T, there's no thymine. Instead, it's replaced by uracil, which is chemically very similar, but it's different enough to have its own name. So, transcription, A, U, A, U, A, U, C, we're going to write a G, C, a G, G, a C, so on and so forth, so that when we get done and we have the information written down in an RNA molecule, you could work backwards and I could tell you what's in the original DNA strand, because the RNA strand is what we call complementary to the DNA strand. Okay, so this information is ultimately determined by this information. Okay, RNA, messenger RNA, remember it goes from the nucleus, there's a little pore, goes out through the pore, out to the cytoplasm, and there the ribosome takes the information in the RNA and it then determines the sequence of these building blocks or amino acids that make up a protein. Okay, so proteins are what we call polymer. They're a large molecule and their subunits are called amino acids. Okay, and the sequence of amino acids in that protein determines what type of protein it is. Okay, and the sequence of amino acids in that protein is determined by the information in the RNA and then ultimately from the information in the DNA. Okay, here's how, again, think about translation. We're now going from this language of U's and G's and A's. We're going to the language of amino acids. So that's, in that way, it's translation. Every time the RNA says U, the ribosome will bring this specific amino acid to the growing protein. Every time there's a G, and a C, then the secret translator in the RNA brings a round amino acid, and so on and so forth. So here we go. U means it's translated into a square amino acid. And every time, here's another U codon, and it tells to bring a square amino acid. So codon turns out the RNA is read in 3 base sequences. Here are those codons. Here's the U. That square amino acid we just saw turns out as phenylalanine, which is a type of amino acid. So if you guys are into your health or fitness, you got those muscle milk drinks. They're filled with different amino acids. Why? Because that's the whole point of working out, is that you're going to tear down your muscle proteins and you're going to build new muscle protein. And what are you building them out of? You're building them out of amino acids. And here are all the amino acids. I think there are 23 different amino acids. 1, 2, 3. No, I can't count because you'll see this again. Don't get caught up in the details on this as a non-major. If you are a bio major, then you eventually probably have to know that CGU means arginine. You guys do not need to know that. What I want you to be able to know here is, again, this is how the translation part of this process happens. If the ribosome reading along the mRNA discovers an AGU, then these rules apply, and an A and a G and a U will cause a serine amino acid to be added to the protein. Okay, so this is the translation step. So a little more detail. Same concept, but shown a little differently. That's how we learn, guys, right? Is you read about it, you see it, you draw it out, you talk about it with somebody else, you see different diagrams, right? Here, the DNA is shown a little bit differently than any of the other diagrams that we've looked at. But you can know that it's DNA. Well, number one, it's labeled. But also, you see, here's the backbone. 2 sides of the backbone. Double helix. Going in the middle. Here's your bases, right? That's where the information is. Okay? So where is this taking place? Well, I see it's DNA. It's got to be in the nucleus, right? Here's the nuclear membrane. Okay, so here's a big enzyme called RNA polymerase. What does it do? This is involved with transcribing the information coded in the DNA into an RNA. And so as the DNA opens, it exposes the bases, where the RNA polymerase adds a complementary base or a complementary nucleotide to this mRNA. So in the end, for this given gene, maybe from here to here, we're going to make a copy of that DNA into RNA. There's some other stuff that happens. It's processed, it's capped, it's spliced, there's SNRPs, blah, blah, blah. Suffice to say, this little thing here, the red with the 2 yellow tails, is a copy of one side of that DNA. And where's it headed? Translation is done. We're now going out to the cytoplasm. We're going to that's right, ribosome. Okay. Again, lots of details. You don't need to know the details here. The information, or the important stuff is, here's the ribosome. Okay. What is the red thing? That is the messenger RNA. Okay. The messenger RNA is going to determine for a given protein coded for by that gene what the sequence of amino acids is. What's this up here? That is the amino acid. Okay. And why did the ribosome bring that specific amino acid? Because of the specific codon on the mRNA. Okay. So remember, we go back to this. Okay. So the 3 letters on the mRNA tell that ribosome to bring a very specific amino acid. Okay. The ribosome moves along. It encounters a new 3-letter codon. It tells the ribosome to bring another specific amino acid. That amino acid is attached onto the protein, right? And as it reads along the mRNA, it just keeps adding more and more amino acids to the protein. Okay, this is complex. This is difficult. I'm telling you guys, you have to read this over in the textbook. You have to, and again, this is right from chapter 4 and chapter 10. Read this over, spend some time with these diagrams, and understand what's going on here. I cannot take this information and fuse it into your brain. I can explain it once. You can watch this video 100 times, but you've got to do the hard work of reading this over and working hard to understand it. Okay, so in the end, what's the whole point? Taking the information, encoded in the DNA, and then using it to make a specific protein. Okay, so we have accomplished that. That's the central dogma of molecular biology. And at the end, I'm going to have to bring this back and swoop back around and make the case that in order to understand cancer, you've got to understand that. Okay, because what is cancer? Cancer is caused, in every case that we know, cancer is caused by this thing called a mutation. Okay, mutation in your DNA is what leads to you developing cancer. Cancer is caused by mutagens, whether it be human papillomavirus or pubic warts, or whether it be pesticides, or whether it be the UV from playing too much golf. Ultimately, cancer is caused by these things called mutations. Okay, and so here's an example of a mutation. When you see this diagram, look at it and say, what am I looking at here? By now, you should be able to make sense of what's in this diagram without me explaining. Don't just make me explain everything to you. I will do my best, but you've got to do the work, right? What are we looking at? A's, U's, G's. This looks familiar, right? Here's that sugar phosphate backbone. What are these? Those are the bases, right? This is, and we know because we see a U in there, this is an mRNA molecule. So underneath that, we've got this MET that matches up with 3 nucleotides on the mRNA. Here's a codon. What is this thing? An amino acid. Methionine, lysine, phenylalanine, glycine, alanine. Right? The sequence of these nucleotides and the bases is what determines the sequence of amino acids in this protein. So here's a normal gene and a normal protein. Okay? What if we take this A, or the G rather, and we substitute it, we flip it? Instead of being a G there, we now have an A. What's the implications of that? So we go from GCC to AGC. GGC? Yeah, GGC to AGC. So when the ribosome is trying to translate the information in the mRNA, instead of going and looking for a GC and bringing a glycine to the table and adding it to the protein, we now go A, G, C, and bring a serine. Okay? So we changed the sequence of amino acids in that specific protein. Okay? So rather than be, and here it is, it's very predictable. It's that in every type of cell that we know about reads mRNA the same way. That's why we can take a gene from a jellyfish and insert it to a mouse, and that mouse's ribosomes will make that protein that makes it glow in the dark. Right? Yeah, it's true. Okay, so that's a mutation, is we're changing the sequence in the DNA, therefore the mRNA, and then ultimately changes the sequence of amino acids in the protein. Okay, so this we call a substitution. Same number of bases in the end, but one of them is swapped for the other. Now, we can also experience what's called a deletion. So we're focusing on this codon here, UUU, and what happens is one of the U's gets deleted, and so what we see is a whole shift. We're losing one of our nucleotides. We're losing one of our bases. And so as the nucleotide or the ribosome reads the mRNA, from this point forward, all of the codons are changed. Because rather than UUU, this G is now shifted. The G that used to be in this codon is shifted to that codon, and we see kind of a boxcar effect. And because there's only 2 nucleotides left at the end here, we actually are missing an entire amino acid. Okay, so a deletion, you can insert a given nucleotide. So same codon, pop in a G, shifts the U over into this codon, and this A gets shifted off. It's not going to be used for anything. So we end up, rather than having this sequence of amino acids, we have almost an entirely different sequence of amino acids. Okay, so it could be you're in the sun and the radiation from the sun breaks your DNA, it changes the sequence of DNA, and therefore rather than making this protein, you make this protein. Okay, and again, the hard part about biology is everybody wants to understand how cancer works, but we've got to labor through these little concepts, right? And they're all building on each other. And so now I hope if you're sitting there and paying attention, you may be asking yourself, well, who cares about the sequence of amino acids in a protein? Right? So we're building up to this. Change the sequence in the DNA, which changes the RNA, and now we know that it changes the protein amino acid sequence. And so the logical question is, who cares? Why is that important? And why does that cause cancer? And why does that cause so much pain and anguish and death? Okay. So what can be the type of things that cause a insertion, a deletion, or a substitution, right? So here's one of the study questions is, know the various types of mutations, and then what are different types of mutagens, and why should you avoid them? So here are some major classes. This is not certainly an exhaustive list of things that are mutagens. A mutagen is a substance or just something that causes a DNA to change its sequence. Okay, so radiation, we know that things like X-rays, things like gamma rays, things that like radioactivity will physically break the bonds between those bases and can change the sequence of the bases in a DNA. UV, we know UV causes cancer. Skin cancer is caused largely by exposure to too much UV radiation. Why? Because it does exactly like we've been looking at these slides. It causes change in the DNA. Chemical mutagens. Of course, we know cigarettes, but it's becoming clear now that there really is no, in terms of mutagenesis and in terms of cancer threat, there is no safe level of alcohol to be consumed, right? That alcohol, even a few drinks a week, raises your risk of breast cancer, colon cancer, stomach cancer by up to 10%, right? So these type of chemicals, as long as they can somehow change the structure of your DNA, can lead to cancer through mutagenesis. And then another one I think a lot of people are sort of surprised by, viruses cause a lot of cancer. Cervical cancer in women, penile cancer in men is basically almost entirely caused by a specific virus called human papillomavirus, which you can be vaccinated against. This is pubic warts, so unsafe sex with too many partners can cause cancer because those viruses get into the cell and they inject their DNA, and that DNA can be incorporated into your cellular DNA and can change or mutate your DNA, which can lead to cancer. Right? So these are different things that can change the sequence of DNA in your cells. So getting back to this question about who cares about the sequence of amino acids in a protein. Okay, this one you could spend your whole life studying proteins and proteomics and the shape of proteins, but I'm going to try to make this relatively simple. On the left, we see a type of protein, or this is an enzyme, and we see it's like a necklace with a bunch of little beads on it. Those beads, guess what they are? Yes, they're amino acids. Okay. Now the sequence of those amino acids, because some amino acids are attracted to each other, some are repelled by each other, because of the chemical properties of the specific type of amino acid, it determines how that chain folds into a protein. Most proteins are not just a long string. They are three-dimensional and they have a specific shape to them. Okay, and so we see on the bottom right here the final folded structure of this protein, and the way it folds up is determined almost entirely by the sequence of amino acids, which you now know is determined by the sequence of the genetic material, those bases in your DNA. Okay, so the sequence of amino acids is important because it ultimately determines the shape of that protein, and the shape of the protein determines its job. Okay, great example of this. Many folks are aware of, at least this, it's really a family of genetic diseases called sickle cell, and particularly folks in the African American community are aware of this because it's one that folks in that community suffer a lot more from, and there's really interesting reasons for that to do with evolution. But if you have this disease, then your blood cells are not shaped correctly. So we see on the right here a normal red blood cell shaped kind of like a lozenge, or round with a nice little indentation. Folks with this genetic disorder have this sickle-shaped red blood cells, and they are not as able to carry oxygen, the sickle-shaped blood cell, that is, and so it basically causes like an anemia, causes a lot of clotting issues, causes a lot of exhaustion, and so how does your genetics at birth determine whether your blood cells are going to be normal shaped or misshapen, whether they're going to be healthy or whether they're going to be diseased, right? It's the DNA in that person's cell, once it's translated into a protein, at this specific, and this is a very well studied system, so they actually know exactly what causes this. But let's see, there's a substitution. So in other words, this cysteine amino acid is changed to a different type of amino acid, which then causes this long chain to fold and be shaped like this rather than like this. And then when a bunch of these polypeptides come together to make the actual cell, the cell is shaped like this. Okay, so the sequence of amino acids determines the shape of the protein. The shape of the protein determines whether that protein works properly or not. And this, by the way, I'm showing you sickle cell anemia here, but this is the same for cystic fibrosis, type 1 diabetes. Any disorder that is caused by genetics is caused by this type of story right here. Okay, very important. Everybody will deal with some type of genetic disorder in their life, in their family, in their friends, and understanding this is, I just feel like this is important. Okay, so then the next question, right, what's the big deal with proteins? Okay, so your proteins aren't shaped correctly, and that I've been sort of hammering on this idea even in 3 lectures. You should have heard me say this multiple times, but a cell is really nothing more than a bunch of proteins that are interacting and doing their jobs. The proteins are what make up things like hair and nails, antlers in the sheep on the bottom. They are storage proteins. So in an egg, the egg white is called albumin. That's what the growing embryo draws its energy out of to build the new body. It takes the protein amino acids from the albumin and builds its body. Proteins involved with movement of our body by contracting actin and myosin in our muscles. They are responsible for movement of cells. Inside of cells, the organelles are moving around on the cytoskeleton. There are proteins that are involved with moving things in and out of cells. They are involved with, it's not too simplistic to say if it happens inside of a cell, it happens because of some protein. Enzymes, whether you can digest milk, whether you can, really any chemistry that happens inside of a cell happens because of an enzyme. Enzymes are proteins that need to be shaped correctly because the genetic code for that protein is correct. Okay, so proteins, and we're going to really just kind of foreshadowing in our story of cancer. Of course, we said cancer is caused by mutations. So we know then the implications of the mutation is that it changes the shape of some protein, and that is what leads to cancer. Okay, so these proteins are also involved with telling a cell whether they should divide or not. Okay, and that's where we're headed. The next couple of lectures, we're going to talk about cell division, one cell becoming two, right? All cells that exist because a pre-existing older cell divided, right? And so when that happens, how fast that happens is determined by protein messengers in a cell, and whether or not or how good a job those proteins do and how many times a cell divides is determined by the shape of the protein, which is determined by the genetic code for that protein. Okay, so probably one of the, I guess, these are a couple of lectures that I think that you have to have some self-discipline to be able to put your nose to the grindstone, read through this idea of transcription, translation, mutation, how it affects proteins. If you can understand these things, you can go a long way towards understanding cancer, and really a lot of evolution, a lot of things in biology. But it's kind of drab at first, and I'm going to just encourage you, I'm going to be your cheerleader to really get you to get down and understand these things, at least at the level like I've discussed here in this lecture. If you will thank yourself later, I promise you. So that's the lecture. I'm going to cut this off here. Next time, we're going to be talking about a little bit, more specifically about chromosomes, DNA, and about cell division, mitosis, and how that is controlled and how that is related to cancer. Okay, so appreciate your attention. You guys hang in there, and we'll talk to you soon.